just keep awake for another 40 minutes or so. Let's go through this, okay? So actually, this is after alignment. This is the first part that goes into the analysis, and this part is not difficult, fortunately, and you don't need a lot of brain cells to be activated to understand it. So it's a straightforward thing, but there's some considerations you have to make when you analyze the data. Okay. So we went through this flow chart that I thought is pretty neat, and this is the part that I'm going to focus, which is the gene expression part. It's very beginning stage. And the question here is, after we got the RNA sequencing data, how are we going to measure the gene expression? And it can be pretty simple, right? So if this is one gene, this is another gene, and this expresses this much, this expresses this much, and you just measure the number, count the number of rays that fall into each individual gene, and you can have a pretty reasonable inference on how much the regional expression level is. Okay? But things become a little bit more complicated with this type of scenario because, as you can see, the longer the gene is, even with much lower expression levels, if you only count the total number of rays, they are the same. So it's not a very reasonable, very accurate measurement. It's only based on the counts, right? So we know that the number of counts that we are detected is determined by a couple of factors. The first one is the level of expression of the gene. So some genes express more than others, and of course, the more it is, the more rays that we'll detect. And the second one is the length of the gene. The longer genes that we have the potential to see more rays. And the third one is actually the depth of the sequencing. So if for one sample, they are based on the same depth of sequencing, but for different samples, and one sample you may use 20 million rays to map the entire transcriptome, the other one you use 50 million rays. And of course, for the second one, that you will detect more signals in the second sample, but that doesn't mean that gene expression level is higher. So these three factors actually determines the number of rays. And the measurement for that is something very simple, and it is first presented in that Nature Method paper just I talk about, the first paper I talk about. It's called RPKM, or it's called reads per kilobase exome model per million mappable rays. Okay? So R here is the expression level of the gene. This is something that we want to estimate based on RNA sequencing data. And the C is the number of total rays that fall into this region. This is our measurement. And the N here is the depth of the sequencing, how many total rays that we are having. And the L is the length of the gene. Okay? So you do this level of normalization. So you can see this measurement is straightforward, meaning that they are doing two levels of normalization here. There is normalized based on the length of the gene. The longer the gene is, the more signal you're supposed to detect, and also based on the total number of depths of that sample. So this is something called RPKM. Okay? And later, that with the pair end rays, and then people talk about FPKM, and it stands for fragments per kilobase exome model per million mappable rays. Basically, they are the same thing, just one for the single end, the other one for the paired end. Okay? One example here, I just made it up. I want to make a point. Here is one gene total exome size, let's say 3,000 base pair. An RNA sequencing experiment contains 32 million mappable rays. We have detected 512 rays in the exome region, and conduct expression level for this in RPKM. So what you do is you got this total number divided by this K, this M, which is 32 million. You just put 32 there, right? And the other one is 3,000 base pair. You just put 3.23. So this is in a unit of 1,000. This is in a unit for million. And after that, you calculate this gene expression. It's close to 5 RPKM. Very simple, straightforward. Yes?
So you would do this?
Each sample, each gene. Yes.
Right.
Yeah, we'll do this for each sample, each gene. And there's a couple of factors that affecting RPKM. This is kind of straightforward, and some of the samples are longer than others, and some of them have higher expressions. So if you only look for the read count, you can see this 1, 2, 3, 4, and this is their numbers. But once you correctly calculate the RPKM, you can see the fourth sample, even though the total count is high, and the expression level estimation for that gene is not very high because it's longer than the other isoforms. It's very straightforward stuff. Okay, but things are more complicated. If it's that easy, we don't have to go through this, right? Especially that we talk about Chris Birch paper, we found out as many as 92% of multi-exon genes can be alternative spliced, meaning they have multiple isoforms. And this is not a this is not an individual problem, it's a universal problem, because we know that in general genes are. And many genes have close paralogs, some reads cannot be assigned into a transcript. So let's look at this figure. So you can see this is one gene have two different isoforms. There are some exons that are included in both, and others are unique to each other. Okay? For this type of scenario, how do we calculate that L? How do we calculate that length? That becomes a tricky issue. And you can see that what we are observing is at this location there are so many rays accumulated, and at each individual location. But how can we reallocate each individual rays back to their proper isoforms? It can be tricky, right? And for these two, we probably can think, oh, let's just use this to measure this isoform 1, use this to measure isoform 2. And those are pretty straightforward. But for the constitutive exons, which means that the exons are in both two isoforms, how do we know that which read is from which isoform? It can be tricky. And the strategies to do that is to estimate isoform level expression rather than using the entire gene as a unit. Now you're using each individual isoform as a unit. And by counting only the reads that map uniquely to a single isoform. So what that tells you is when I estimate the expression level for isoform number 2, I only use these rays. Okay? For isoform number 1, I only use these rays. And all the other rays, I cannot use it because I don't know which isoform it comes from. The problem of this approach is it loses too much information because we know that alternative splicing occurred a lot, but the alternative spliced exons are still only a very small part of the gene. So that we are not using the whole information. And they're also not working for genes without a unique exon. Sometimes if you have multiple isoforms, there's a crazy thing. There's one gene in Drosophila has over 30,000 isoforms. Okay? It's bigger than the entire human genome. So once that thing happens, you can forget about that. There's no way we can study that gene anyways. But the point is that there is a lot of genes, they have more than 3, more than 4 different isoforms. Those are pretty common. But how do we reallocate those back? It becomes really, really tricky. And the next strategy is better. It's kind of called isoform expression methods. And what they do is that they use the maximum likelihood methods, which I'm not going to go to the equation part of it, maybe a little bit in the next lecture. But the point here is, and actually example include the cufflinks and meso are both general. I mean, the people in my lab knows these two very intensively. I mean, they know that we use that a lot. But the point is, this is some type of isoform expression methods. Okay, and the general strategy for that is I'm going to start from those rays that are unique to each individual isoform. I'm going to estimate this. But that is only my point estimation, meaning that I don't have a confidence interval. I don't know what's the range of it. Okay, and then they further use a statistical approach to reallocate the number of different rays in the constitutive exon into different isoforms. Okay, and this is more like a statistical inference issue. And at this stage, you not only get one measurement for the percentage of the isoform, how many was the percentage expressed in the final product, but also you are getting a confidence interval of that. Okay, so based on this approach, it's not only using it use more information in the exons that are uniquely assigned to each individual isoform, but also it takes advantage of the other isoforms in the same time, other exons in the same time. So that increases the powers. And this type of approach is not accurate for low expression genes with many different isoforms. Okay? And but things doesn't have to be that complicated, right? So if our goal is to estimate gene expression per gene, not per isoform, I don't care about isoform. I just want to know how much this gene expressed, right? Alternative splicing, which isoform expressed how much, that's other people's business. So let's first define a gene's expression, and which is the sum of the expression of all its isoforms. Okay, straightforward. And if we are going to this route, there's still two general approaches. The first one is it's called the exon union method. So let's go through this figure a little bit. And this is for the first isoform and this second isoform. These are all on the transcript level. And this is on the genome level, so different exons. So you can see if I see a blue, dark blue one, that means it's only coming from this, the first isoform. This is only from the second isoform. And for this hybridized ones, it is from both two isoforms. Okay? For the exon union method is, I'm going to focus on everything as long as it present, this exon present in one of all the isoforms, I'm counting it. This is my overall length of this particular gene. And on the contrary, there's another one. It's called exon intersection methods, which means I only focus on the exons that occurred in both two isoforms and getting rid of the middle ones, which are alternatively spliced. Okay? There are two general strategies. For the exon union approach, which means when I'm using the entire, as long as it occurred in any of them, I'm going to use it. Okay, there's a problem of that, which is the underestimated expression levels for alternative spliced genes. Okay, so what actually, if you think it through, it's quite simple. And because once I see this particular exon and expressing in the second isoform, not in the first isoform, but when I make the calculation using the everything together as my length, I'm over-counting the length, so I'm over-correcting my samples. And here is in the real sample, you can see that this X is the true value, and this is the estimated value based on this approach. And this exon union method clearly underestimated the expression level of the gene. So that is a potential problem. Okay? And for the exon intersection method, which means I only use the constitutive exons, and for the alternative splice ones, I get rid of it. And the problem of that is also reduce the power of differential to identify differential expression, because you are getting rid of a lot of information which may be useful for some other applications, and so you're supposed to have better, more deeper measurement, but you don't get it because you remove those alternative splice exons. Okay, this is another thing that I don't expect you to read from the board, but the point is we also have all the code to achieve this type of functionalities to calculate the RPKM based on whatever alignment file that you may get. Okay, our code also includes diverse functionalities. It includes gene expression, alternative splicing, and count rates on predefined regions. And it's a flexible gene model, which means that today we got another paper that have non-coding RNA and new annotation. We can chip in our new annotation into that, and we can calculate the things that we are calculating pretty easily. And it also has the flexible normalization schemes. So the total the M, the RPKM, the M normalization, which is the total number of arrays, that we can also use all arrays in the region, or all arrays in the genome, or 75% of arrays in the genome. So there's things that you can tweaking around. So different groups, they are using different strategies, but our code can do that. Okay? The last topic of today, maybe 5-6 slides, is about RNA sequencing differential expression. So the previous section, we are talking about quantification. We are talking about which gene express how much based on RNA sequencing data. We are not talking about comparing two different samples. Now we are getting to the summary of how to do the RNA sequencing differential expression analysis. So the fundamental question here is how the expression level differ across conditions. Sometimes we are interested in just quantify it. Sometimes we want to see which gene differentially expressed between normal and cancer samples, for example. And over the past decades, actually, many methods have been developed for microarray-based studies, right? And you can use t-tests, you can use more deeper analysis tests based on different type of normalization. But actually, many such methods can be directly to the RNA sequencing data. For some earlier studies, and that we can use the RNA sequencing data, calculate RPKM values, let's say full samples in one condition, full other samples in another condition, and for each individual gene, we just do the t-test. Well, that works, right? Especially when you go back to do the validation and if it works. But that is not using the full potential of the data. However, that can work. So using read coverage to quantify the gene expression levels, the RPKM, things that we talked about in the previous session, and then do the various statistics based on that. Okay? The distributions for the expression estimates needs to be considered, because when we do the t-test, one of the major assumption is the data follows normal distribution, right? And sometimes it doesn't. And for the arrays, once we do the log transformation, we see the data roughly follows normal distribution. For the RNA sequencing, probably it's not. And if you know the specific distribution, make better sense, and you can build that into your estimation model. Okay? And the power of detection of this differential expression depends on the depth of the sample, how many arrays you devoted for each individual sample, the expression of the gene, and the length of the gene. For multiple factors are important. And to detect the differential expression, the first approach that being used is the Fisher's exact test statistics. It's very, very simple. So if you think this is one gene, and these are the arrays I'm detecting in one sample, this is in another sample, okay? And here is I detect 16 arrays out of this 10 million overall arrays fall into this region, and here is 10 arrays versus 16 out of 3 million overall arrays. So it's not that difficult to see which gene, this gene, in which sample this gene expressed more than the other, right? It's clearly the bottom one express more because the total number of overall arrays is much less than this, and this is not too much less arrays following to this region. Does that make sense? Okay? And the way to do that becomes very simple. You just form this 2x2 contingency table. Okay? If you took 651, the introduction of biostatistics, then you know this is the most basic approach in the statistics. So you see, just give the number of arrays in sample A overlapping with the gene 16, not overlapping is 10 million minus 16. And so is for the second sample. And then you do the Fisher's exact test, you will be able to figure out whether this gene is differentially expressed or not. You have a question? No? I thought you were thinking. Okay. And, all right. And for the fold change, how much different the gene expression level is, and you can calculate the fold-out ratio for that. It's very, very basic stuff. And for this type of approach, for experiment with biological replicates, And what people do is combine the samples. And this is really a waste because you are not taking advantage of the distribution of the data at all. Okay? You just merge it together. And a better approach for that is to do the t-statistics on a generalized linear model-based gene expression estimation. Okay? I don't want to be very mathematical driven, but this formula is kind of pretty simple. So you can see that this is your measurements. Okay? We know the measurement is determined by the lane effect, and which is the total rate is one of the effects, and maybe some other effects. And it's kind of a batch effect type of thing. And this is the gene expression levels. This is something we want to estimate. And this is other type of specific errors. So what people do is once they establish this type of linear, generalized linear model, and you will be able to estimate this value, which is the gene expression based on the total number of counts. And then you further do the t-statistics on that. So that is people's general strategy. But still, it's not really optimal because when we do the statistical inference, if you think of t-test, the most simple case, right? And what you really need is two levels of information. The first one is the value, the differences. Okay, so our point estimation. And the second one is the variation. And if this is not very accurately measured, the variation is bigger. Otherwise, it's tighter. And if it's tighter, you'll have a better chance to give a good p-value. So you need not only the difference you measured, which is the point estimation, but also you want to understand the variability. What's the distribution of the variability, including the technical variability and the biological variability in these samples, in a large number of biological attributes. And there is a good assumption for that is the read counts follow to one gene region actually can follow a Poisson distribution. Okay? And if you think of the original definition of a Poisson distribution, I mean, it's in a limited time, the number of events occurred, or things like, it's very much similar to what we are observing here for the RNA sequencing data. For a big chunk of genetic region, and how many reads fall into that region, that follows the Poisson distribution, okay? And you can see this is the equation for that, but the major advantage of assuming it's Poisson distribution is we have a good estimation on the variance. Because if it's a Poisson, that means that the variance is equal to mean. Okay? If we can estimate our mean based on the observation, and we can roughly have an idea of the variance on that particular sample. So you got not only the mean, but also the variance, and that is making your statistical inference much more powerful. Okay? Are we following this? I hope so, remotely. Go through the podcast if you did not, okay? Well, but you can see that for the Poisson distribution here, I'm having a question mark here. I mean, in theory, in principle, it makes sense. We are assuming the data follows the Poisson distribution. But in reality, is that correct or not? Okay? But once we go to people look at the real data, they found it's not perfectly correct. There's over-dispersion factors are involved. The so-called over-dispersion is when observed variance is higher than the theoretical variance. Okay, here is the theoretical variance. Okay, it's equal to the mean. But when people look at the real data, they found for the larger expressed genes, and their variance is higher than the mean of that time. So this is called the over-dispersion. So in order to fix this problem, when over-dispersion factor is considered, Poisson distribution can be generalized into another distribution called the negative binomial distribution. Okay, I'm sure that a lot of you know the equation of that. And honestly speaking, I've never seen it. All right, I know I can find it in Wikipedia, but it's not going to be very difficult. It's be more difficult than that. But the major difference is it not only have the two parameters that are needed here, and it has another factor correcting the over-dispersion. Okay? If that over-dispersion factor is equal to 0, Poisson distribution is equivalent to, negative binomial distribution is equivalent to Poisson distribution. What happened? Okay. Oh, yeah. So there's a lot of other things, statistical considerations that can be done, and there's pages of equations you can look at, and all these things are in these dots, right? And the good news for that is some people know that a lot of people are not capable of understanding what they're talking about, so they made our package on that, right? So there's a couple of statistical packages in the bioconductor package, or some of them are in the established softwares, and EdgeR is one of it. Actually, it built in 5 different assumptions, different models into it. You just run through it and let me know what it looks like. I haven't tried it. Okay. And all right, another point here is when we talk about a differential expression between 2 different conditions, okay, we are assuming that our quantification is accurate. So when we estimate the gene expression levels, we are assuming we already know their gene expression levels. So we want to only talk about the differential expression at this stage. And but as we know that it's not that trivial, it's not that simple, right? There's multiple ways to do that. So just pay attention to that. So this is one example of quantification strategies can affect the differential expression analysis. Okay, intersection method didn't detect anything for this. So if I only use, this is one sample, this is another sample, the read distribution. So if I only use the constitutive exons, and your end result is you don't see the differential expression between these 2 isoforms at all. Okay? And if I'm using a union approach, which is the overall thing, I'm going to see this slightly difference, and therefore there will be potentially a significant p-value. So you can see exactly the same data, same analysis. I'm just using 2 different ways to quantify my gene expression, and the result can be so different. Okay, you've got to pay attention to that. And actually, if you use the isoform expression method for this, meaning that not using the entire gene as a unit, rather to separate that into multiple isoforms, and you will be able to get a much higher sensitivity on the detection. However, we know that those methods are pretty tricky, and sometimes I don't really, they gave a good p-value, very good p-value. When I go to look at the data, they just don't look like right. So when you see the risk distribution, sometimes you have to do the balance. But the point here is the quantification can affect the gene expression difference detection. Summary of this part is the capacity of identifying differential expression can be affected by multiple factors. This includes sequencing depth, library preparation, flow cell effects, and GC contents. And the good news is more and more bioinformatics statistical methods are being developed at this stage. And if you look at the bioinformatics papers, it will come out, something come out every day. Okay, I think that's the end of it. For the next lecture, we will have 2 parts. The first part will be a guest lecture and talking about transcriptome profiling in tumors. And I will finish that up with alternative splicing study and gene fusion identification. All right? That's it. Have a nice weekend.